Hello everyone. Let's get started. Today we're going to be talking about translation. If you recall from the central dogma, translation is the process where a cell uses messenger RNA to build a protein. Recall that all proteins begin as polypeptides, long chains of amino acids bound together with peptide bonds. And that once the polypeptide is created in the cell, it will be carefully folded into a three-dimensional functional protein. Across all domains of living things, translation occurs at a ribosome. Remember that ribosomes are found in all living cells, and they are located in the cytoplasm. Remember that in eukaryotic cells, ribosomes stud the outside of the membrane of the endoplasmic reticulum, which is why it's called the rough endoplasmic reticulum. In prokaryotic cells, there is no endoplasmic reticulum. There are no organelles. So the ribosomes are free-floating in the cytoplasm. The other thing we need to recall is the genetic code. Remember that the genetic code is essentially a table of all possible combinations of the 4 letters of the bases that we find in messenger RNA, and in fact in DNA. The genetic code tells us what the code in those bases translates to. In other words, which codons match which amino acids that are placed into proteins. The genetic code is the same for all living organisms. The process of translation, in fact, is very similar among the domains. We're going to talk about it as it occurs in bacteria, but in reality, the process in bacteria, archaea, and eukarya is quite similar. There are some important differences in eukaryotic organisms, and I'll point those out as we go today. So, using the bacterial cell as our model, the ribosomes that are in the cytoplasm are going to attach to the messenger RNA as it is being produced. Those ribosomes will then start the process of translation. They will start synthesizing protein before the entire messenger RNA is even completed. We've talked about this before. In prokaryotic cells, transcription and translation occur simultaneously. As genes are transcribed and mRNA is produced, ribosomes will attach to the mRNA and begin translating it immediately before it's even completed. It's a different process in a eukaryotic cell, remember, because eukaryotes have organelles, including a nucleus. The process of transcription in a eukaryotic cell takes place in the nucleus. So the entire messenger RNA will be produced and in fact processed into a mature transcript before it is sent out into the cytoplasm for translation. So these are sequential processes in a eukaryotic cell. Transcription happens first, and then translation happens. We say that those two things occur consecutively. On this slide at the bottom, you can see a drawing of this concurrent process of transcription and translation in a prokaryotic cell. If you recall, this simultaneous transcription translation produces what are called polyribosomes in the cell. In other words, strands of mRNA that are being produced through transcription that are attached to multiple ribosomes. It's drawn for us on the bottom part of the slide, And then at the top part of the slide, we can actually see an electron micrograph that has been colorized to show us these polyribosomes emerging off of the DNA molecule. So here's the strand of DNA. Here's a strand of DNA. You can see that a particular gene or perhaps an operon is being transcribed. It's not being transcribed once. We're not going to make one messenger RNA. We're going to make several messenger RNAs from that gene. And that's why you see multiple strands of mRNA being produced. And as they're being produced, the ribosomes, shown in red, are attaching and translating. As soon as they can possibly get onto the mRNA, they do. And just like we said a minute ago, that multiple mRNA will be produced from one gene, the same is true for translation. Multiple copies of the protein will be produced from each mRNA. On this slide, we can see a depiction of a eukaryotic cell that's been cut in half so we can peek inside. And this is to remind us of this important difference that happens in a eukaryotic cell, which is that transcription and translation are separated in time. You can see the cell membrane here around the outer edge of the cell, and then they've indicated for us where the nucleus is, right here. We see the depiction of the DNA molecule in red, and we can see that the RNA polymerase enzyme in green is transcribing a region of the DNA, a gene. You can just make out that an mRNA is being produced. It's shown in yellow. That mRNA will be produced in its entirety. Remember, in a eukaryote, that molecule is not ready for translation. It has to be processed. We talked about how splicing has to occur. The introns have to be removed from the mRNA, and the exons have to be attached to each other. Then the molecule has to have a cap and a tail attached to it. Now it's considered a mature mRNA, and it's ready for translation. It's going to exit the nucleus through one of the nuclear pores, and now ribosomes can attach. In the drawing, they're only showing us one ribosome attaching to the mRNA, but remember, multiple ribosomes are going to translate this mRNA. We're going to get multiple copies of the protein that this mRNA codes for during translation. As long as that mRNA exists, it will be translated. Recall that mRNA don't live very long, so we will get multiple copies, but that mRNA will then be fairly rapidly degraded. You can see this green strand emerging from the ribosome. This represents the polypeptide, the chain of amino acids that's being produced. Once that is complete, it will be carefully folded up to produce a functional protein. So, translation not only involves the messenger RNA, but it also involves the two other forms of RNA that we've talked about, ribosomal RNA, which is part of the ribosome, and transfer RNA, or tRNA. We haven't really talked about tRNA yet, but it plays a major role in translation. The first thing that has to happen, in fact, for translation to occur is that individual tRNA molecules have to be loaded with individual amino acids, because it's the tRNA that is going to help deliver the correct amino acid for each of the codons in the mRNA. It will be the job of the ribosome to expose one codon at a time on the mRNA, and then only one tRNA will be able to come in and interact with that codon. There will be one tRNA that has the correct anticodon on it that will match in a complementary manner with the codon and deliver the correct amino acid. The amino acid on that tRNA will be transferred. It will be taken off of that tRNA and it will be bound to the growing chain of amino acids that is emerging from the ribosome. So, the very first thing we need to talk about in terms of the actual process of translation is how these tRNA molecules get loaded up with their amino acids, and how exactly those amino acids get taken off of the tRNA and added to the growing chain. Over here on the right-hand side of this slide, you can see a drawing of the process of translation in its entirety. They're showing us the mRNA molecule as this black strand, and they've depicted the individual bases of the mRNA as these sort of little teeth jumping up off of that strand. So all of these little projections represent the A's and the U's and the G's and the C's that are in the mRNA. This gray molecule here represents the ribosome. Remember, there's a small subunit and there's a large subunit. The ribosome is literally going to travel down the length of the mRNA molecule, and it's going to expose one codon at a time, one group of 3 bases at a time. Those bases can only interact with one type of tRNA, and that's the tRNA shown here in black that has the complementary anticodon on it. So if there's a C in the codon, we're looking for a tRNA that has a G in its anticodon in that position. If there's a U in the codon next, we're looking for a tRNA that has an A in its anticodon in that position. It has to be a match. There has to be a complementary match between the codon on the mRNA and the anticodon on the tRNA. If there's a match, then whatever amino acid that tRNA is carrying is going to get placed at that position. So how are amino acids loaded onto tRNAs? Well, it's probably no surprise to you at this point, but there's an enzyme involved in that. There's an enzyme called amino acid tRNA synthetase. And it's this particular enzyme that is going to perform a process that we refer to as activating the tRNA. This process requires energy, and it's going to come in the form of ATP. So in the middle of the slide, you can see basically the formula for activating a tRNA. An individual tRNA molecule and the appropriate amino acid for that tRNA are going to come together through the action of amino acyl tRNA synthetase and energy provided by ATP to create a combined tRNA amino acid molecule called an amino acyl tRNA. This is an active form of the tRNA. In other words, this is a tRNA that is now ready to participate in translation. We refer to this process, we refer to this reaction that's performed by the amino acyl tRNA synthetase enzyme as charging the tRNA. We're charging it up. We're getting it ready to do its job. So An important thing to note here is that there are different tRNAs for each of the 20 amino acids that we find in proteins. That means that there has to be a different enzyme for each one of these charging reactions. Remember what we've said about enzymes. Most enzymes, the vast majority of enzymes, do one reaction. So there has to be a different amino acyl tRNA synthetase for each of the different tRNAs. It's amazing the assortment of enzymes that are required for this. So the tRNA and the amino acid that it is intended to carry, according to its anticodon, and the enzyme, and ATP, are going to produce an active form of the tRNA, an amino acyl tRNA. That's the term we use. This is actually a two-step process, interestingly enough. The enzyme is first going to combine with the amino acid. So amino acyl tRNA synthetase attaches with the amino acid, and then that molecule, the amino acid enzyme complex, will then interact with the tRNA and will end up with the charged amino acyl tRNA molecule. I just want to point out here that when we think about these things, it's important that we keep scale in mind. So if you look at this drawing, we're looking at a charged tRNA. We're looking at an amino acyl tRNA. So this large molecule shown in green, this is the enzyme. This is amino acyl tRNA synthetase. And all these little green circles that you see, these represent individual amino acids in this protein, because that's what an enzyme is. It's a protein. So this molecule over here, the one that looks like a 7 or an upside down L, recall that this is the mature tertiary structure of tRNA. tRNA is just one of the forms of RNA that we see in a molecule. And it, just like all RNA, it goes from its primary structure, which is a sequence of bases, into its secondary structure, which is the hairpin, remember? Into its tertiary structure, which for tRNA is this molecule that looks a little bit, again, like the number 7. So here's the tRNA in comparison to the enzyme. And here's the amino acid it's carrying. Remember, each one of these little circles represents an amino acid. So the one in pink is just another amino acid. And it happens to be carried by this particular tRNA. And that means that if the amino acid is carried on this end of the molecule, the anticodon is on this end of the molecule. There's a group of 3 bases down here, which is going to interact with a codon, but only one codon, or the correct codon, if you will.